Hey everyone, thank you for attending my talk today. I'm Masa. I work at Purdue University as a postdoc and in Dr. Linda Lee's group. Today, I will talk about the PFAS remediation with some reactive nanocomposites. And before that, I won't go through the details of why we care about PFAS, but I will have a brief summary of what has been done in terms of the removal. So far, the most common way to remove PFAS from the water is through the sorption process that usually sorbents like granular activated carbon or different sorbents are used. But the main problem with these techniques is that they are usually sequestering PFAS, and there is a likelihood of re-release and also a waste that has been contaminated with PFAS that needs to be taken care of. And on the other hand, we have destructive techniques like electrochemical oxidation, UV-generated hydrated electrons, but most of these techniques are very energy intensive, or they're not amenable for use in situ, and also they do not address all PFAS. Then we have zero valent iron, which is a strong reductant. It's environmentally friendly, and it has shown promising results in terms of PFAS removal, but the main problem is it should be in subcritical water, and there is a tendency for nanoparticles aggregation. But if we add some catalysts like nickel and we add some support like activated carbon, we can overcome the problems that we can have with only zero valent iron. That's what we did, and this part of work has been already published if you're interested in more details. So they are not. So what we did, we synthesized nickel iron on activated carbon, and these nanocomposites were able to mineralize and defluorinate PFAS up to 94%, and that was accounting for all PFAS transfer. So this really motivated us to look further, and we looked at the effect of structure, and which I'm not going to show here in terms of time saving. And we also looked at the effect of this and how this technique can be used as in situ. So here is the proposed way that we think it can be applied. So the main idea is if we have a groundwater plume contaminated with PFAS, these particles can be injected as a permeable reactive barrier, or they can be on like above the ground and as an injection. So basically, instead of having activated carbon, the GAC, we will have a reactive GAC that can be used to remove PFAS while not only sequestering it, but also destroying it. And this is the overall proposed way that we think it can work. So with this in mind, we have this technical approach where we did column studies. So the column was the glass column. They were used glass column, and they were packed with sand as support. And also we had the reaction bed that had both sand and the nanocomposites in it. Then we looked at the PFAS as organic and also the inorganic fluoride in both effluents and also in the extracts of column after sacrificing column that the period of experiment was done. And then we looked at the targeted PFAS that we introduced into the system. We also did suspect organic products that we were expecting to see from PFAA transformation by using a high-res mass spec QTOF. And then this is the column study set up at Purdue University. As you see, we had different pumps for different flow rates that we used to pump PFAA through the column. So we looked at the pH and ORP in the effluent, the subsamples of the effluent, and also we had some inline measurements for both pH and ORP. And these work all were done under oxygen-free conditions in a chamber, but the column was taken out from the chamber and the reaction or the experiment was started in the lab. So we did 4 different column studies. 2 column studies were focused on only PFAS. They were the preliminary studies we did, and they were both at 60 degrees C with different flow rates. Then we had the column studies with mixtures at 2 different temperatures. And from the results we had from batch studies, we tried to optimize the condition for the last column studies we performed, where we decrease flow rate to increase the reaction time. We optimize the reaction bed to have more nanocomposites ratio compared to sand. And we had PFAA mixture, but at a lower temperature. For one of the scenarios, we buffered the PFAA mixture to see if that will affect the pH profile or not. and also the reactions to see if the buffering will help the reactions or not. So here I'm showing the pH and ORP measurements in both inline measurements and also in the effluents. And I should say that the pH profile really varied by the reaction, by flow rate, and the temperature. But overall, for 3 studies out of 4, we had an increasing trend in the profile of the pH. And as you see, for the one that was actually buffered, that increased, we didn't have that much of fluctuations, and it was a nice increase the whole time. For ORP, we also had the similar observations for both inline and also the effluent subsamples, and the increasing trend was consistent between these 2 observations. So with that, when we were looking at the column, and if you want to see the PFAA transformation in the column, we needed to see how each PFAA was actually retarded in the column. So we get the data from our batch studies, and we looked at the KD values to see how much of PFAS will end up in the material of the column, while how much will go to the pore volume. It was an assessment, and since we didn't see any longer chains ending up in the supernatant or in the water phase, that's why we extrapolated, and we have a nice linear correlation. But since the nanocomposites was playing a much more important role than sand in retardation, that's why we didn't look at the KD values of sand for other PFAS, and we just used the extrapolation. Then we used the KD values, and we estimated the retardation factors for each PFAA based on the mass of the sand versus nanocomposites and their representative KD values. And with that, we were able to estimate the reaction type of each PFAA in column studies. So here I'm showing the results for the single column PFAS studies at 60 degrees C at 2 different flow rates. And we got similar results where about 60 to 75 to 80% of PFAS was removed from the system. PFAS never broke through the column even after 105 pore volumes. However, we found PFAS in the extracts, and as expected, PFAS was only recovered in the inlet of the column, which shows that PFAS never made through the layers through the exit of the column, and that's why it never actually broke through. So it was all sequestered and got reacted in the first layers or in the reaction bed. And here we are seeing the breakthrough of different PFAA through the column. So we have mixed column 1 and mixed 2, where for mixed 1, we have a higher flow rate, and that's why we collected more pore volumes. And as you see, the PFAA breakthrough actually follows the expected trends for both mobility and the reactivity. Shorter chains actually break through earlier, and they have shorter residence times, which also translate to shorter reaction times. And the reactivity, and as we see here, the breakthrough is also linked to the reactivity, which makes sense because we also observed higher reactions of longer chains in batch studies that I will show in the next slide. So here I have the results for the batch studies that were performed for 5 days. As you see, the percentage recovered, or the percentage not recovered, which means percentage transformed, is increasing by the chain. So as we have longer chains, we have more percentage of PFAS transform. And this was a mixture study, and I also have the single PFAA as diamond. As you see, less is recovered for the single PFAA, which means that more is transformed. And again, what we see in the column is very consistent what we saw in the batch studies. Here I am showing the PFAA recoveries from different layers of the column. And as you may remember from breakthrough of the different PFAAs, longer chains never broke through the column. And that's why we see P4, PFNA, and C8s only residing in L1 and L2, which is the inlet layers, while the shorter chains made through the whole column, and they are found in all the layers that they were also found in the breakthrough. They broke through the column. And this is very consistent with the single column studies that we saw PFAS was only residing in layer 1 and 2. Here I will have this is a very comprehensive slide that shows what we found for column studies and also batch studies. So if we look at the mix 1 versus mix 2, mix 1 is in green bars while mix 2 is in pink bars. So we had more transformed in mix 2 column, which had lower flow rate, higher reaction time, better optimized reaction bed at 50 degrees C. When we look at all the data together, you will see the circles are showing the PFAA mixture batch for 5 days. And we have more recovered in those, or we have less transformed. But we should consider that we had much higher reaction time in the columns versus the batch reactions, which were only for 5 days. And we also have the batch study for Cape Cod, a real groundwater contaminated with PFAS. And we had the lower transformation with that, which makes sense that can be linked to the other water quality parameters present in the groundwater. Also, I have the single diamond bold as a PFAS column. And as you see, among those two columns had very similar flow rate, and we see similar percentage transformation. Overall, we have a higher percentage of transformation in mix 2 column. When we looked at the fluorine recovered, we have 73% recovered for mix 1, while only 55% for mix 2, and we have better fluoride generation in the column with higher flow rate at 60 degrees C. However, this can be linked to the higher temperature that can help in higher mineralization. Also, it should be noted that we looked at the suspected organic products that now I'm not showing it here because that's whole another thing. And it can really account for the fluorine mole balance that is missing here, because usually if we estimate the concentration of those organic products from our experience in the lab, we will underestimate it. So when we see much higher organic products, role in the mix 2 as expected. So overall, when we look at the whole data together, we should see that the higher reaction time actually ended up in higher percentage transfer.
About 3 minutes left.
So here I have the slide that shows the percentage not recovered or the transformed of different PFAS and which nicely is correlated to the number of fluoroalkyl carbons, and it is very consistent with what we observed in the batch reactions. And there is a nice correlation for both columns and the higher percentage overall for mix 2 compared to mix 1. So with that, I have this summary slide, and I think with this technique, we're able to have to remediate PFAS or remove PFAS under 1D flow condition. And what's expected is different PFAA will have different affinity to sorb to the particles or to the reaction bed of the column, while they will break through the column following their Bose mobility and the reactivity effects. And I should also add that we were able to see the inorganic fluoride as the main product in all the effluents, and that we self-sampled and we analyzed. And I would like to acknowledge my PI, Dr. Linda Lee at Purdue University, Dr. Choi and Zenobia for their help, and also our funding resources. So with that, I think I'll get any questions that you have.
Thank you very much. Yeah, we do have time for one question.
Give a minute to see if anyone has any. How is, oh my goodness, something with the iron? Do you see that?
Yeah, I got the question. I saw it. So the nano-nickel iron activated carbon particles are synthesized in the lab, but there are companies that they are interested in our particles, and that's something we can do in larger scale, I should add. But this is basically a synthesizing based on the combining them. So nickel iron, then they are combined, and then they are synthesized and activated carbon by storing for 3 hours under oxygen-free condition, and then those particles are ready to go. So that's the basic synthesis of the particles.